This whole event is the final in a series of events. It's been sponsored by a grant from the Templeton Foundation to Jennifer Doudna and Bill Hurlbut, who will be moderating the last panel, so you'll meet him soon. And we just want to thank the Templeton Foundation for their support in this very important topic. At the IGI, we're a life sciences institute, but we also recognize the importance of weighing the way the science fits into society. And for that, we're conducting research, and we're committed to public engagement and stakeholder engagement on these topics. And what topic is more important at the current moment than editing the germline? So what we're doing here today is not so much a referendum on yes or no, but really trying to address some of these bigger questions about how do we involve stakeholders and publics in these big conversations. So to get this started, I am going to hand it over to Ben Hurlbut, who's going to tell you a little bit more about that charge, what we're trying to accomplish today. And then we'll get everything started.
So thank you very much to all of you who are here today. Many of you have come from far and wide, so we are most grateful for your participation. I'm just going to say a few quick words of framing to get us started. So as Lee mentioned, the sort of occasion for gathering us together here are the development of very powerful tools of genome editing, and in particular a tool called CRISPR-Cas9, where foundational work for the development of that tool was done here at the IGI. This technology allows targeted genetic modifications of just about any being from microbe to mammal. There are myriad extraordinary, promising, and hugely valuable applications of this technology. And of course, there are also those that are ethically fraught. And perhaps amongst the most controversial among these is the potential to modify human gametes and embryos to introduce genetic changes into children, heritable genetic changes to the germline that would be passed on to not just to those children, but to those children's children and on down through the generations. So this ability to make genetic changes to the DNA of future children raises far-reaching ethical questions. Should it be done? For what purposes and subject to what limitations? But just as crucially, who should decide these questions? How should we as a human community guide and govern these emerging technologies? A few years ago, a group of leading thinkers convened an international summit on human gene editing and concluded that we should not proceed until there is, as they put it, quote, broad societal consensus about the appropriateness of its use and for what purposes. Those words have caused a certain amount of controversy, not least because there's uncertainty about what such a consensus would look like, and a skepticism that it could ever be achieved, hence the question mark next to consensus in the title of this gathering. These are both important questions, and yet seeking such a consensus, or at least engaging in the kind of broad deliberation that might move us in that direction, is surely a worthy aspiration. And it's in that spirit that we convene this conversation, to contribute in a small way to a larger imperative, a larger effort to develop sort of broader, more expansive and more inclusive dialogue that draws upon the breadth of human experience and moral imagination. And we're facing real challenges. Scientific and technological advances are proceeding often without guidance from wider society about what technological futures we want and what futures we don't want. As I'm sure everyone in the room knows, last year a scientist in China created the first genetically engineered children. His aim was apparently to race ahead of his scientific competitors, but also to reshape and speed up, as he put it, the ethical debate. But speed is surely not what we need in this case. We need thinking and reflection and deliberation informed by the fullest range of cultural and moral and religious perspectives. After all, the human future is at stake. And yet, to date, most of the discussions that have informed governance approaches surrounding genome editing have played out in expert committees and summits that have engaged a relatively limited range of perspectives. These have been worthy and important efforts, and yet insufficient and incomplete. Surely expert input plays an absolutely crucial role in governance. But as multiple expert committees who have convened these conversations have repeatedly affirmed, expert judgment is insufficient. We need a broader, more inclusive dialogue. But questions remain. How should public debate proceed? Through what mechanisms and processes? Who should lead and shape it? And even where it's taking place, how should scientific and technological agendas and governance approaches be tethered to public deliberation and judgment, lest one or a few reckless scientists take the reins from the rest of us? So our task today is to confront these challenges and explore possible paths forward. We have the privilege of engaging in a dialogue about how to guide and govern these emerging technological possibilities through a dynamic and quite diverse group of participants in a public conversation that will draw upon the expertise of a range of interdisciplinary, international range of people from around the world. But also that we hope and we intended would include a much broader community, in effect, anyone who wished to accept the invitation to come and participate in the dialogue. So this was meant to be a very public affair with a much larger gathering of the public in the spirit of the project of this meeting. Unfortunately, we had a little bit of a lesson in who has the power to shape debate, and it turns out it's the power company. So I think as everyone in the room knows, and people who are tuning in from afar maybe don't, the local power company Pacific Gas and Electric has deemed today to be the day that it will turn off electricity to the UC Berkeley campus. So we were supposed to have gathered in a much larger room. There were roughly 200 people, members of the public, signed up to come and participate in this discussion. So it's a sort of shame and irony and even absurdity that we've had to exclude that public that this gathering was intended to include. And yet, nevertheless, fortunately, the web remains with us. What would we do without it? And so, at least judging from our numbers of the people who signed up, there are another sort of 250 people or so who promised to tune in. And we hope you are with us. We encourage you to stay with us. There are mechanisms that I think Lee will say a bit more about for participating in the discussion, mediated, unfortunately, by the technological means of participation. We would much rather that there were more people in the room, but so it goes. And so we are really going to rely upon you, those of you who are out there in cyberspace, to participate and contribute to our dialogue. One other quick thing. So we were one of the sort of, there are multiple complex, interwoven, choreographed elements to this meeting, all of which have been profoundly disrupted by PG&E. But nevertheless, one of the elements of this meeting is a collaboration between the organizers and Tomorrow Life, which is an emerging project to create a kind of online video repository that will kind of capture and contribute to conversations in this domain. And so we'll hear a little bit more about that from the leader of the project, Samira Kiani, during session 3. But there is a film crew here who was meant to capture the conversations both among the invited experts and among the public that would unfold in the coffee breaks in our big and open space. And so we're going to sort of play it by ear, what they will do and how they will do it. But nevertheless, they are here. There are cameras floating around. And insofar as you have thoughts that you want to share, conversations that you would invite others to listen in on, to eavesdrop on, we invite you to find that camera crew, or if they come up and approach you and stick the camera in your face, to welcome it, and recognize that the things that are said here are in the spirit of sort of building collective dialogue, and that this platform can serve as a means to invite more people into the space of conversation. By the way, if you haven't noticed the releases, the notices that are posted around saying, watch out, you're on candid camera, that probably dates me, but anyway, they're there. So please take note that anyone who's here may land on film, and if you have concerns about that, please do make them known to the film crew and to the organizers. Finally, I want to reiterate some thank yous first of all, to our sponsors and to the co-organizing and hosting institutions, but also to the range of people who've been involved in pulling this thing together. And I'm just going to read off the names because each of these people have put in crucial efforts. Megan Hochstrasser, Kevin Dozen, Stephen Crane, Dana Carroll, William Hurlbutt, Lori Perko, Christy Nordhall, Yulia Glubovskaya. How did I do? Anyway, sorry, Yulia. Susan Jenkins, and then most importantly, my co-organizers, Julie Shapiro from the Keystone Policy Center, and Lee Witkowski, whose incredible heroic efforts to salvage this thing over the last 12 hours are really the only reason that we're all here and not, dispersed wandering around doing our own thing. So with that, I'll turn it back to Lee.
So before we get into the first session, we wanted to kind of bring everybody up to speed on the science that we're talking about here today. It's going to be very minimal. There's actually not a lot of science that you need to know to be able to participate in this conversation. But we want to make sure that the terminology that you might hear today is something that you recognize and are familiar with. So to get started, let's quickly go over some basics of what is this genome editing thing that we're talking about. So there's an analogy that I think is very useful to use. You can think of a genome as a book of instructions, and it has all the information needed to create an organism. It is organized within that book into chapters and sentences, or you can think of that as chromosomes and genes. Genes are the sentences that contain a full individual instruction about one part of creating the organism. That terminology in the book, the language, is in DNA chemical bases. So you may hear some terms like genome, chromosome, gene, DNA. That's what we're talking about today. And the editing mechanism, there's many different sorts. The kind that we're probably going to talk about most today is CRISPR. It's the new exciting version, but there are also different ones, and I'm sure there will be more to come. The idea with genome editing is that you can actually target that sentence in the book that you want to target and make some modifications to it. So if there's a mistake in the way it's written and you have a mutation, you can target it, identify it, and correct it, sort of like a find and replace function, if we were to translate this from a paper book into an electronic version. So that's the general concept, is this ability to kind of rewrite the instruction manual. And beyond that, there's some other terminology that you might hear, like somatic cells and germline cells. So the thing that we're focusing on today is this concept of rewriting the instruction manual, the genome, of germline cells. The distinction that we're drawing here is one that is very useful, but it's also one that you might hear challenged today. It's one that I think we should consider a little bit further. But to define them, somatic cells essentially are the cells in your body of an existing person that have often been differentiated to be things like skin cells, bone marrow cells, adult stem cells, so they can also be created, encouraged to divide and become new types of cells, but these are cells that cannot create a new person by themselves. Germline cells, on the other hand, are things like eggs, sperms, and embryos, and the progenitor cells that create those. And so the distinction here is that if you modify the genome of a somatic cell, that modification will stay with that one person that you made the modification in. Whereas if you modify a germline cell, that modification will be passed down both to the person that will be created from those germline cells as well as to all of their descendants. So there's something a little bit more profound about modifying the germline, and that's what we're here to talk about today. As you can imagine, this is a very controversial topic. So you are going to hear lots of different worldviews, lots of different perspectives and takes on it. We are welcoming all of those here today. So in order to have a productive conversation, we have some of these guidelines for how to engage this topic. So be empathetic. Assume that even if you disagree, the person is approaching this with good intent. Be curious, dare to listen, so dare to try and understand where somebody else is coming from. Engage fully, disagree respectfully, and dare to disagree. So we want to hear different perspectives. Share the floor. So when you do come up to the microphone and you want to ask a question, please be mindful that there are others that might also want to share the floor with you. Create common understanding. So even though I just defined some terms, let's do our best to avoid some technical jargon so that we can speak to everybody. And let the MC, me, Lee Witkowski, and the moderators, we'll have a timekeeper sitting over here, do their jobs. So we're here to spark conversation. So there's one other group that I wanted, in the spirit of engaging different worldviews and different perspectives. We've heard a lot of recognition and thanks for the organizers and for the Institute for East Asian Studies, which is hosting us today. It very kindly offered their facilities. But I also want to recognize that Berkeley sits on the territory of the Huichin, and I'm hoping I'm pronouncing that, the ancestral and unceded land of the Ohlone people. This land was and continues to be of great importance to the Ohlone people, and we recognize that every member here at Berkeley and the community has continued to benefit from the use of this land. So consistent with our values here of community and diversity, we wanted to affirm Indigenous sovereignty and acknowledge and make visible the university's relationship to Native peoples. So to get started, we are going to be using something called Poll Everywhere. And if you imagine this big auditorium where we would have had all these people sitting in the audience, this would have been a wonderful way to get everybody sharing their perspectives. We're going to do that still because we want to hear from those of you that were able to come today. And there is a version on YouTube that's being posted where you all can join us and input your opinions and your perspectives. So the question is, what issues drew you to this conversation today? So let's set kind of the frame of why everybody came here today to discuss this topic. And you'll see that some of the words start popping up. So we're seeing technology, change the world, ethics, future, access, equal dialogue, stakeholders, power, change, eugenics, groundbreaking, dialogue. Ooh, yeah, now we're getting somewhere. Lots of words. Ethics is coming to the fore. All right, justice, publics, generations, inclusion, children. So the big ones right now are dialogue, ethics, and society. So that is exactly appropriate, I think, for today. So let's move on to one more to get a sense of who all came into the room with us today. So here's the next question. With what field or study? So we know that not everybody here is academic, but what are you most familiar with? What sort of knowledge do you most identify with in this category? So we're seeing lots of life scientists. That's great. I'm glad you're all here to have this conversation with us. Social scientists and humanities, and other. That will be really interesting to find out what other is later. And we'll be posting the results of this after the conference is over, so you can all look. So this first session, this first panel, is about the stakes and stakeholders. So before we jump into that, let's get a sense of what everybody in the room sees as the stakes when we're talking about governance of germline editing. So in 1 to 2 words, what are the stakes that we're here to talk about in your perspective? So the major themes here are future, humanity, equity, trust, power. Many of the same things that everyone answered of why they came to the room today. These are The things that are at stake is why you came. Humanity. All right. All right, so let's go to the next question, if it's working. So what issues in human health and reproduction need to be informed by the public? So again, today we're here to talk about the engagement aspect of governing this, not as much about should we or shouldn't we, but how do we make that decision and how do we involve the appropriate people? So what issues do you see as being needed to be informed by the public? Human welfare, everything, access, all cures. Who decides? Boundaries. Just give it a couple more seconds to see what else comes in. Patient voices, economics. All, everything, I think. Lots of alls. All the issues in human health need to be informed by the public. Access issues. The IGI poll needs to be. Yeah. Humanity, irreversible change. What counts as a disease? Definition of disease. Disability justice. Understanding of human health should be informed by the public. Transparency. Everything. All right. Eugenics should be informed by the public. Getting rid of politics. It's a big ask there. Policy for applied research. Autonomy. All right. Great. So let's go to the next one. So that was what should be informed by the public. So what issues in human health and reproduction should be reserved for experts to decide? Bioinformatics, nothing. Safety, eugenics, science. Science should be reserved for the experts. Safety should be reserved for the experts. Limits, safety. What goes on in the test tube is the domain of experts. Technical risks, nothing, none. Technical feasibility, safety, and efficacy. Public education should be reserved for experts. Collateral damage. Scientific details. None. How do we define experts? Experts get to define experts. Tech evolution. Common values. Methods. Whether or not to have strawberry cream cheese at lab meeting. Boundaries. Nothing. Methods. All right, so that's how the poll everywhere works. And we'll do that at the beginning of every session to kind of get a sense of where everybody is in the room. And we'll come back to this at the end of the session.